Hey guys, so in the previous video we talked about Mendel and his experiments and continuous and discontinuous variation and the pea plant and what he was looking at in the pea plant. So let's talk about those laws of inheritance, lots of terminology, and we are going to play around with inheritance in this video. So terminology that we need to know. We talked about dominant and recessive. Those are alleles. What are alleles? Alleles are traits. Traits like brown eyes, blue eyes, dark hair, light hair, fair skin, dark skin. Those are all traits controlled by your genes. And alleles are going to be different versions of those traits controlled by your genes. So if something is dominant, we're going to write it a certain way when we're writing our Punnett squares. Hopefully you've heard about Punnett squares. Hopefully you've done Punnett squares before you came to school here, before you've done them in college. I mean, in high school before you got here. But just in case, we're going to refresh all about Punnett squares because that's what we're going to do in this video. So alleles that are dominant, we're going to write them as a capital letter. And in this case, I'm just going to use A, but substitute whatever letter you want to as your dominant letter. Recessives, we talk about recessive traits that they are masked by the dominant ones. So dominants are going to be capital letters, and recessives, when we draw them, they're going to be lowercase letters like that. So recessives are masked by dominants. We have to have both traits of them to be able to show them. What am I talking about when we have both of something? If we have two of the exact same allele, it's called homozygous because homo means same. Homo means same. So homozygous, if we were going to write something as homozygous dominant, and we said that dominant is the capital letter A, then homozygous dominant, homo, I'm going to abbreviate it, homozygous dominant, we would write as two capital letters there as homozygous dominant. What if something was homozygous recessive? It'd be the exact same, except for homozygous recessive would be lower cases that are the same instead of upper cases that are the same. So homozygous, remember, homo is the same, heterozygous is different. So if we were going to write heterozygous, we would write this as one dominant and one recessive. That's heterozygous. You may also hear heterozygous referred to as being a carrier. You're a carrier of something. Usually means that you have one trait that's one thing, one allele that's one thing, and one gene that is carrying for something else. So you're heterozygous. Other terms that we need to talk about are genotype and phenotype. Genotype and phenotype. So we're going to mention this and then we're going to show different examples of this later on. But a genotype is going to be simply the genes that there are. If you looked at these genes that we just wrote down with our homozygous dominant as two capital letters, homozygous recessive as both lowercase letters, and then heterozygous as one uppercase and one lowercase letter, we have three totally different genotypes. If we were going to write genotypes for these, genotypes, it would be three different ones. A ratio of one to this one is different to this one is different. Those would be the genotypes. But we said that if you have a dominant trait, that trait would mask the recessive one. So if we looked at these three sets, if we looked at this one, this one, this one, and we said A is, I don't know, dark hair. We'll say A is dark hair. And lowercase a is fair hair, light colored hair. If we looked at the homozygous dominant and we said that capital letter A's were dark hair, then two capital letter A's are definitely going to give you a phenotype of dark hair. If we looked at the homozygous recessive and they had two lowercase a's, that's going to be fair hair that shows up. And what do we think about the heterozygous? Heterozygous has one dominant and one recessive. It has a dominant in there. So if it's heterozygous with a dominant trait, it's going to show whatever that dominant trait is. And in this case, this would show up as dark hair. So if the phenotypes are what show up, what gets expressed, what you're actually going to see. With a phenotype ratio, we would say that it would be 2 darks to 1 fair hair, a 2-to-1 ratio, if we were looking at phenotypes. So just laying it out there. Genotypes are the genes that you have. They are what they are. So the genes are the genotype. The phenotype is what actually shows up. Dark hair, A blood type, whatever that dominant trait is, what are you seeing? What are you looking at? What is getting expressed? So that is the difference between genotypes and phenotypes. Genes are what they are, what they are, what they are. Phenotypes are what are you seeing? What are you looking at? What is getting expressed? Phenotypes. And sometimes phenotypes will differ in their ratio number than genotypes. So we're going to see this later on. We talk about genotypes and phenotypes. Okay, phenotype. If we looked at these guys, we have yellow and we have green. So yellow and green parents in our parent generation, or the P generation. When we combine these two parents, we said that F1 generation is all hybrids. So before we looked at purple and white flowers, and now we're looking at yellow and green peas, and all of the offspring in the F1, that first generation of kids, they're all yellow. And then we're going to take the F1 and breed it with other F1s. We're going to take 2 of those progeny from the F1 generation. We're going to breed those 2 together. And when we breed those 2 hybrids together, that's going to end up with our F2 generation. And we end up with 3 yellows and 1 green, or a 75-25 split out of the group, or a 3-to-1 ratio, 3 yellows to 1 green little p. So these are the phenotypes. The phenotype would be written as a 3-to-1 ratio for phenotypes. Our genotypes are going to be different though. So we look at these guys, and here we swap sides. I don't know why they swap sides in this picture, but this guy is our green and this one is our yellow. We're looking as far as genotypes. So we swapped them, green and yellow, but we end up with the same amount of F1s. Everyone in F1 is a hybrid, and those genes are written like this because you're going to have 1 gene from mom and 1 gene from dad, and everyone in the F1 generation is going to have this genetic makeup of 1 capital and 1 lowercase. If it was written as 1 capital and 1 lowercase, what do we call that? Is it homozygous or is it heterozygous? If we have 2 different ones, they are heterozygous. So we have a heterozygous, nothing but hybrids here in this F1 generation. Every genotype is the same. It's all the same. Let's breed those F1s together. So we bred the F1s, 2 F1s together, and we end up with the second generation, or the F2s. On the F2s, here's how those genotypes showed up when we did this. We end up with some that are homozygous dominant, 2 capital letter Ys. We have some that are homozygous recessive, and they're going to have both lowercase y's. And then we're going to have some, 2 of them to be specific, that are that hybrid, just like the parents were genotype. So in this case, we have a 1, and there's 2 here, and there's 1 here. So we would write this as a 1-to-2-to-1 ratio for our homozygous dominant, homozygous recessives, and our heterozygous there, a 1-to-2-to-1 ratio as far as genotype. So our genotypic ratio and our phenotypic ratio were different. Genes are genes, they are what they are. Phenotype is what gets expressed. So even though we have a 1-to-2-to-1 genotype ratio, both of these guys still express as a yellow color, while only this one lone guy expresses as a green color. So let's keep playing around with this kind of strict dominant recessive kind of traits, because P's are a great model for this system. Dominant traits, other dominant traits. We've been talking about pea plants and their dominant traits, but humans are going to have dominant traits and recessive traits too that we will talk about. I'm not going to ask you to remember all of these guys. Like achondroplasia is a dominant disorder that affects your not your bones, your, why am I blanking? Cartilage. Oh my goodness, your cartilage, which is going to affect your long bones. because your long bones, as you grow, have that growth plate, and that's made of cartilage. So chondroplasia is going to affect that. It's a dominant disorder. Brachydactyly is going to be, most of us are going to have 5 fingers, but some of us have 6 fingers. We have an extra finger, and that's brachydactyly. Huntington's is going to be a neurodegenerative disorder that we'll talk about in just a little bit. Marfan syndrome, neurofibromatosis, widow's peak, right there, widow's peak. And woolly hair are all dominant traits, meaning they only need one. Only need one. They only need one. They only need one gene to show up or to be expressed. And they will mask the recessive one if there is one. If there's both dominant ones, then it is what it is always. But even if it is heterozygous, you're still going to get that dominant trait show up. Recessive traits, albinism, where you're lacking your pigment. CF, cystic fibrosis, where you're missing you're not missing mucus. You have a hard time controlling your mucous membranes. It's a genetic disorder. It is inherited, and you end up with excess mucus production in all of the areas that are mucous membranes, respiratory, digestive, repro. All of these things have mucous membranes, and that can cause trouble with this excess mucus production with CF. Muscular dystrophy, we're going to talk about that when we talk about sex-linked disorders later on. Galactosemia, PKU, these are all kind of digestive, being able to break down different things. Sickle cell anemia is going to be where you are building up the proteins to make hemoglobin. And when you do this, the amino acid gets switched out. It switches out one amino acid, and that ends up with sickle cell anemia. Tay-Sachs is going to be a pretty much lethal genetic disorder where people don't make it past very young ages, like 3, with Tay-Sachs. And all of these are recessive. So with recessive, you need both. Remember, recessives are going to be lowercase, dominants are uppercase. Recessive, you need both genes to express. So you need to have both of these, while these only need one for a dominant trait. So we said Huntington's was a dominant. It is a lethal disorder. Most of the time when you're thinking that this is a dominant trait and it's lethal, how is it even getting passed on? So unfortunately with Huntington's, it is a dominantly lethal disorder, but it does not display until later in life, maybe in your 20s, maybe in your 30s. And by then you might have already had kids and you didn't know that you had this neurodegenerative disorder, and you've already passed it on to your children, unfortunately, because if you have passed on this trait, again, it only takes one gene, and you might have already passed it on and not even known that you did. But once symptoms do start showing with Huntington's disease, it is 100% fatal. It is neurodegenerative. And I think life expectancy is about 12 or 15 years after it starts to show up. So this is an example of an autosomal dominant where you just need one gene of this thing to display and be expressed and show up. Other recessives, I don't know why the name for this, but this is cystic fibrosis. This is Tay-Sachs, which is a lethal disorder where children barely make it past the age of 3. And PKU is where you have a hard time digesting phenylalanine and you cannot handle that in your diet. So now we test every baby to see if this baby has PKU or not, because you can adjust the dietary intake of that amino acid and it'll be just fine. It'll be just fine. But you need to know ahead of time. So those toxins don't build up and cause potential retardation in growth for baby. Okay, so let's play around with some of these: strict dominant and recessive. Another type of disorder that is recessive is albinism, where somebody is lacking pigment. So this is a strict dominant recessive. How do you set up strict dominant recessives? Punnett squares, we are gonna walk you through it. So Punnett squares, I love Punnett squares. I think they're fun to figure out all the different possibilities of something. So in this case, we are gonna deal with, excuse me, oh, cystic fibrosis, right? Isn't that what we're dealing with? Where is it? Yes. Okay. So we're going to talk about cystic fibrosis. And this was a recessive disorder, cystic fibrosis, CF. So we have a carrier father and a carrier mother in this situation. And with these carrier traits, they are going to write them in a Punnett square, we would write somebody who's a carrier as heterozygous. They are heterozygous. So in this case, for Punnett squares, we have mom, and we have her on the top, and we're going to write her genes right here. And we've made our box, and we're going to put one of mom's chromosomes here. Mom has a big capital C for a dominant, and then she's going to have a lowercase c for the CF gene that she is a carrier for. She does not display this because she is a carrier. She is not actually displaying and expressing cystic fibrosis. Same for dad. We have dad on the side, and dad's chromosome. Dad is going to have one normal C, and then dad is going to have one lowercase c for cystic fibrosis. He is also a carrier because they told us over here that dad was a carrier and mom was a carrier. So we have dad over here and we have mom on the top. You can totally flip-flop these. It does not matter who you put on top of your Punnett square. If you put mom on top or dad on top, you're going to get the same result. So what do we do with these? So we're going to bring down, I like to start with mom. I always put mom on top of my Punnett squares. You can do however you want, but I'm going to start with mom, and we're going to bring down her genes. So mom has, remember, one capital letter C and one lowercase letter c because she is a carrier. So let's bring down that capital letter C, and we're going to put it in both boxes. And then we're going to bring down on the other side. She has a lowercase c. So we're going to bring down the lowercase c and put it on both sides. So those are mom's genes. Mom's genes are good to go in the box. Let's bring across dad's genes. Dad's genes, we are going to put, I'm jumping, but they're going to go there. And there we go. There's dad's genes. And we're going to bring those across. Dad, we're also going to bring, again, this one and here, dad's genes. So we brought across dad's capital letter C and dad's lowercase letter c into both boxes all the way across. And what we end up with is two big C's here, one big C, one little c, one big C, one little c, and two little c's. So we are going to end up with, if you have a big C, like this box, and this box, these big C's again were normal, big C, normal, little c is cystic fibrosis. These three, this one, and this one, all are going to show up and express as normal. These are going to be normal, or cystic fibrosis is not expressed. So 3 of them, or 75%, are normal. This last one over here, this one is 2 C's. Both have the recessive gene, which means that that recessive gene will be expressed. If you are homozygous recessive, you are going to express whatever that gene is. And in this case, that gene is cystic fibrosis. So our ratio is 3 to 1, or 75% normal, and 25% are going to express for cystic fibrosis. So if we had parents that were both carriers, their likelihood of their offspring having CF, expressing cystic fibrosis, is a 1 in 4 chance, or a 25% chance of expressing cystic fibrosis, of them having a child that has cystic fibrosis. A 1 in 4 chance, a 25% chance with those parents with carriers. What about albinism? How about this one? So let's set up a Punnett square for this one. We could say, in this case, that mom, we'll say mom, we'll say mom's a carrier because it looks like mom has normal pigmentation. And we're going to say a normal, and again, normal in the generic sense because none of us are ever normal. But A would be normal pigmentation, and lowercase a, capital A is normal pigmentation, lowercase a would be albinism, where you are lacking the melanin in your skin. So we're going to say, I'm going to say this is mom and this is dad. Again, it doesn't matter which one you put on which side. I'm going to put mom on top. And I'm going to say mom is a carrier because I can see in this picture that mom has normal pigmentation. So with her being a carrier, she would be heterozygous, and I would write her just like this: one capital and one lowercase letter. She is heterozygous to be a carrier. And let's say dad is normal pigmentation. If dad has normal pigmentation, then he would have both capital letters, right? So dad would have A and A. What happens if we get a cross between this? If we get a mom that's a carrier and dad that has normal pigmentation, what do we get? Let's see. So I'm going to pull moms down. Mom is here and here. Mom is here and here. And I'm going to pull dad's across. Dad, capital letter, and then the second one, capital letter, capital letter. Knowing what we know about dominant and recessive, we're looking at these 3 boxes. How many in these boxes are going to be normal, are going to have normal pigmentation? This one, normal. This one has a capital letter, also normal, normal, and normal. So this is 100% chance of having a normal expression child. There is a 50% chance that their child will be a carrier, but the likelihood of their child actually having albinism, 0% chance of that happening. This child with these parents, no matter what, will have normal pigmentation. They may be a carrier or they may be homozygous dominant. Let's see if we had mom was a carrier and dad was a carrier. So let's try a new one. We could do it different. We'll do mom over here and dad over here. If mom was a carrier, we'd write her as an uppercase and a lowercase, right? She's heterozygous. If dad is a carrier, uppercase and lowercase because he's heterozygous. And then let's bring the genes down and across. A for dad, lowercase a for dad. Capital A for mom, lowercase a for mom. All right, so with these parents both being carriers, what is the likelihood that they would have a child with albinism? And in this case, this would be normal because we have a capital letter. This is normal because we have a capital letter. Normal because we have that capital letter for dominant. This, 1 in 4 or a 25% chance, 25% chance of albinism. In this combination with these kind of parents. So you could do this kind of stuff all day. You could play around with this kind of stuff all day. This is strict, dominant, and recessive in a Punnett square. I always like to bring this up because this picture in the textbook that they're telling us about is telling us about dominant recessive traits. And it's saying that freckles are going to be a dominant trait, while red hair is going to be recessive. And so I have my oldest child here, and let's talk about how we have the red hair color, because every time we go out as a family together, and here is a picture from 2 years ago, 3 years ago, 2018, 3 years ago. We took a cruise and we took a family picture, and you can see that my hair is dark now. It used to be blonde, but it's dark now, and my husband's hair is dark as dark can be. It's always dark. So we always go out and in public, somebody's always going to look at my children and say, where did that red hair come from? Where did the red hair come from? How did you get that red hair? I get it all the time. So let's talk about how he got the red hair, shall we? Let's make a Punnett square. So I am going to write my Punnett square, and I'm going to put me on top. Here's mom. And we're going to put dad over here. Dad. And let's talk about family history, because when I was giving birth and they, popped my little son on my chest and they were like, "Oh, look at that red hair." I wasn't even thinking about it. And I probably should have been thinking about it, but I wasn't. It didn't even enter my picture to think about that my kid could have red hair. But I should have, because my husband's father, my father-in-law, has red hair. He absolutely has red hair. And my mom and my dad, they both have red in their hair. They are not redheads, but they do have, my mom's strawberry blonde and my dad has a little red mixed into his hair. You can definitely see it in his beard, little bits of red in there. So how can we get a child with red hair? We have to have hybrid parents. That's the only way, right? So let's write that down. I am going to say, again, we'll say that um, big A was dark hair, right? And little a is going to be fair hair. And we're going to say, in this case, it's red. Also, bleach blonde, towheads blonde, 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 blonde is also recessive, just like red hair is. So in order for us, parents with these genes, to have a redheaded child, let's do the Punnett square. So I'm going to write mom and I'm going to bring it down. Mom's capital letter, my dominant A, and my recessive A. Dad's dominant A, and dad's recessive A. Now, looking at these boxes, what's the likelihood that parents with these genes are going to give birth to a child with red hair? Boom, here it is. It's a 1 in 4 or a 25% chance that child would have red hair. So there he is. Here is my adorable, he's much older now. He is, oh my gosh, guys, he's going to be 15 soon. Boggles the mind. So he's gonna be driving soon, but here he is in his precious, handsome little self, and you can see his red hair. So it was a 25% chance of us having a child with red hair, and we did. So that is the likelihood. You will see and need to set up your own Punnett squares. You'll need to understand how to do this with strict dominant recessive. You will be told on a test, mom has this and dad has this, and you will need to know how to write a Punnett square. So please contact me if these Punnett squares don't make sense. If you don't understand how to do them, you don't understand how to interpret the results. If I had asked you about these genes and I asked you what is the likelihood of these parents producing a child with dark hair, what if I asked about the dark hair? What percentage would that be? That would be these boxes, right? Because all of these 3 boxes have a capital letter in them, a dominant trait. So the likelihood of dark hair is 75%. So we have 1 redhead and we have 2, Charlotte's hair is a little bit lighter now, but both of these guys were kind of sandy blondes, dishwater blondes, when they were younger. So that's genetics for you. That is strict dominant recessive that we talk about in this section. Again, you will need to know how to write a Punnett square, how to set it up, how to know how to interpret those results. So please reach out if you need help on those. Why do we know how to write the Punnett squares? How do we know how to split the genes? Why is that? Where did this come from? There are 2 laws that help us reinforce these concepts that Mendel first brought to light. The first one is the law of segregation, and that is what we learned when we learned about mitosis, right? We learned about mitosis was replication of a cell, and part of the replication of the cell was replication of DNA in interphase, specifically the S phase of interphase was where DNA replicated. And so we said that due to the law of segregation, that these paired genes have to separate equally into gametes. So there's a equal chance of either gamete receiving the allele. So here we have our law of segregation, and we're going to have where we copied everything, right, an interphase, and then we're going to go through our prophase, metaphase, telophase, and then here's going to be our daughter cells, thanks to the law of segregation. We are going to have random segregation. So we could do this one of two ways. We could say, mom would be blue chromosome and dad could be red chromosome because I like to go against the stereotypes. So dad is going to have the reddish chromosomes and mom's going to have blue. And because of random segregation, there's an equal chance of these things splitting off. Um, so in this phase, in this version of meiosis, the blue goes, both blues, the long blue one and the short blue chromosome, both of these guys, the long and the shorts, all the blues end up in one here. And then that means that all the red ones are going to end up over here. And then we are going to split those guys apart, and the gametes are perfectly blue over here, and these gametes are perfectly red over here. But it's totally random, right? They're random. How they line up in the middle for mitosis during metaphase, what gene lines up on what side of that plate is totally random. So it could very well just as likely be this condition here where we are going to pull the long and the short chromosome and put them into the gametes, but the long and the short chromosome are different colors. So maybe we have one of mom's genes here and one of dad's genes here for the short one. And we end up having this one with a long blue and a short red over here on this side. And over here, we're going to have a long red and a short blue over here. This was dad's long chromosome and mom's short chromosome. So because of this law of random segregation, those gametes, those little chromosomes are going to segregate equally and randomly into the gametes. So everybody has an equal chance of getting these different genes. What happens if you don't know what the genes are? If you have an offspring and you don't know what the genes are, but you can see its phenotype, then we will perform what is called a test cross. A test cross. If you know what the phenotype is, but you don't know what the genes are, perform a test cross. So we have this yellow pea. Here is our yellow pea. And we know it's yellow. And because we already talked about these genes, right? We talked about Mendel's traits and we know it's dominant and recessive in Mendel's traits. We know that if it's yellow, that one of those chromosomes is the capital Y because that was the dominant trait. So we know for certain that at least one of these is the dominant Y, but we don't know what the second chromosome is. What was the second one that came from mom or dad? Did that second chromosome, was it a capital letter or was it a lowercase? We don't know. So we're going to perform the test cross and figure it out. And we perform a test cross with a recessive. So here's our recessive green. Remember, we know that if it's recessive, it needs both traits. We got to have mom and dad genes both together for recessive to display. So we're going to combine this yellow one that's unknown with a known green genotype. And so when we do, here is our green. So we got our lowercase y's over here. And then here is our unknown. We don't know what this is, but we're going to perform the test cross, and if the genes are two homozygous dominant, if they're both dominant, if they're homozygous dominant, then these are the genotypes that we will get. And all of the offspring with this pairing of this Y and this Y, all of the offspring would be hybrids. Every single one in the F1 generation would be yellow. But maybe when we perform this test cross, we are going to perform our yellow P crossed with the green P, and that outcome ends up giving us this and this. Here's our capital Y and our lowercase y, so these are always going to be yellow because we got the one dominant gene. But what if the unknown second chromosome for that yellow P was a recessive? What if it was recessive? It was recessive that we would end up with two lowercase y's. So depending on what the offspring are, is the F1 all yellow or is the F1 a 2 or a 2 to 2 or a 1 to 1 ratio? That's how we know. So if we're not sure what the genes are of something, we're going to cross it with the known genes of the recessive, because recessive are always homozygous recessive. And we are going to perform a test cross, and based on those the offspring, what's the F1 look like? Is the F1 all hybrids or the F1 split? And we have a 1-to-1 ratio on those. If we have a 1-to-1 ratio, then we know that that was a heterozygous hybrid crossed yellow that we used in the test cross. If all the offspring were um, hybrids, that's the word I'm looking for, if all the offspring were hybrids in the F1, then we know that that yellow was a homozygous dominant. So if you don't know what you're looking at, perform a test cross with something that you do know what the genes are, a homozygous recessive. Here is a monohybrid cross with our strict dominant recessives. We are going to have F1. We said, here's our parents. They're both homozygous dominant. Here's our homozygous recessive. We end up with a heterozygous F1. We're going to breed the F1s together for dominant recessive. And when we breed those F1s together, we end up with the F2 generation. And in the F2 generation, we have a 3-to-1 ratio of yellow to green. Our phenotype ratio is 3 yellows to 1 green. Our genotype ratio, though, remember, genes are genes. They are what they are. So homozygous dominance, there was 1. Homozygous recessive, there was 1. And there were 2 of the 4 were hybrids. They were heterozygous. So genotypic ratio is a 1 to 1, while a phenotypic ratio is a 3 to 1. Another law that you need to know is the law of independent assortment. The law of independent assortment. And that's going to say that one gene does not influence the other. Just because I have dark eyes doesn't mean that I'm going to have dark hair too. Those genes aren't going to influence the other based off the law of independent assortment. It should be every possible combination of alleles has the same likelihood of passing on. And again, this is one of those where I'm like, oh yeah, here's this blanket rule of the law of independent assortment. There are exceptions. We're going to get to those later. But based on the law of independent assortment, genes don't influence each other, and every potential combination of genes are going to be used. So here we are looking at 2 traits instead of just one allele of color. We're also going to look at the shape of the P. So here we have a yellow, go back, we have a yellow P. And that yellow pea, excuse me, is also round, a yellow round pea. So both of those traits are homozygous. Those are dominant. So homozygous dominant, homozygous dominant, homozygous dominant yellow, 2 capital letter Y's. Homozygous dominant round pea shape are 2 capital letter R's for round. Now, here's our little homozygous recessive. And this little P, we already said green was recessive, but wrinkly and not smooth and round, wrinkly is also recessive. So this is homozygous recessive with the lowercase y's, homozygous recessive with the lowercase r's. And we will put these 2 together. And when you look for, instead of just one trait, yellow or green, or one trait, round and wrinkled, we are now looking at 2 traits, and 2 traits is a dihybrid cross, a dihybrid cross. So here we have all of the genes put together, so all the different genotypes. We have a yellow and round, we have a green and round, a yellow and wrinkled, and a green and wrinkled. And we're going to do those same things over here. And then we will look at what do we get? What's our F1 generation look like? I'm sorry, here's our F1 generation. What does if we breed the 2 F1s together, what does our F2 generation look like? And so when we did this and we looked at this dihybrid cross, we got a lot of different genotypes. When it comes down to phenotypes, we ended up with a 9 to 3 to 3 to 1 ratio. 9 of them were round and yellow. 3 of them were round and the recessive green. 3 of them were yellow, the dominant color, but the recessive wrinkle. And then only 1 of them was just like the grandparent, and green and wrinkled. So a 9-3-3-1 ratio on a dihybrid cross. Am I going to ask you to do a dihybrid cross on the test? Nope. No, that's just too much. But I will ask you to set up a monohybrid, normal, regular Punnett square. You will have to know how to do this. Okay, next video, we're going to talk about all the exceptions to the rules in the next one.